joining, everyone. For those of you who don't know me, I'm Donald Gardner. I'm the Deputy Director of the Hub for Sustainable Crop Protection, based in the Centre for Hort Science and located in the QBP. I'd like to kick off by just acknowledging with an acknowledgement of country. I'd like to acknowledge the traditional owners and the custodianship of the lands on which we meet today. I pay respect to their ancestors and their descendants who continue cultural and spiritual connections to country. We recognise their valuable contributions to Australia and global society. Just before we kick off with the seminar, it's scheduled from now until 1 p.m. We'll have Q&A at the end of the session, and that can be done via the Q&A button on the bottom of the screen and not the chat button. So today we have the pleasure of hearing from Dr. Anne Sawyer. Anne is an Advanced Queensland Industry Research Fellow, and she's involved in developing RNAi-based crop protection technologies against plant pathogens. She is a UQ graduate. She did a PhD in the IMB on the regulation of photosynthetic gene expression in microalgae, and then a postdoc at Ruhr-University Bochum. She came back to Australia and to UQ in 2018 to work with Professor Carroll and Professor Nina Minner on RNAi-based plant protection. She works closely with partners at DAF, Scion New Zealand, Nufarm Australia, Greenlife Industry Australia, Australian Pineapples, Howe Farming Enterprises, Australian Native Products, and the Australian Tea Tree Industry Association. So that probably involves a lot of emails with lots and lots of stakeholders. So just a reminder, at the end we'll have Q&A, and now I'll hand over to Anne to tell us all about RNAi for sustainable plant protection. Thanks, Anne.
Thanks, Don. So I'd also like to acknowledge the traditional owners and their custodianship of the lands on which we meet today, and pay my respect to their ancestors and their descendants. So today I'll be giving you an update on some of the work I've been doing as part of my Advanced Queensland Research Fellowship, which, as Don mentioned, is on developing an RNA interference-based protection strategy for plants. So I'm specifically focusing on fungal pathogens as well as oomycete pathogens. And I'm doing this with a number of different industry partners. And I'm based both at the School of Chemistry and Molecular Biosciences and at QAFI. I'll just see if I can. Yeah. All right, so you're probably all aware of our current outlook, but I'll just give you an update. It's expected that we'll reach 9.8 billion people by 2050. And to maintain global food security, we're going to need a 70% increase in our food production. And with the current amount of arable land we have, this will require a 60 to 110% increase in agricultural productivity. And we're seeing increased pressure from plant pests and pathogens, and this is also partially driven by climate change. So traditionally, plant diseases have been controlled through the application of pesticides and through genetic modification of the host. However, both of these procedures have some issues associated with them. So when we apply pesticides, it can result in the pests and pathogens developing resistance. There's chemical residues left on our produce, runoff of chemicals into our environment, into our waterways, and there's just a lack of specificity. So this means that sometimes beneficial organisms can also be targeted. And it's actually very expensive, and it takes a lot of time to develop a new chemical. So it can take about 10 years at a cost of $250 million. So new chemicals are just not forthcoming. And the genetic modification approach does overcome some of the issues with pesticides. It's more environmentally friendly. However, there are major issues with community acceptance. There are other environmental impacts. Regulation can be a big issue. There are lots of hurdles to overcome. And not all crops can be transformed, so it's not actually possible for everything. And it's also quite expensive, and it takes a long time to develop a new GM crop. So about 13 years of research and development at a cost of $136 million. And so RNA-based biopesticides has emerged as a new technique that can be used to sustainably control pests and pathogens. And so this was first demonstrated against plant viruses. So I've shown a virus example here. And it's quite easy. What you do is you design and synthesize double-stranded RNA that's specific to the pest or pathogen that you're trying to target. You apply it to the plant. And then when that pest or pathogen comes to invade the plant, this RNA interference mechanism is triggered. And this is a natural defense mechanism present in a lot of eukaryotes, which basically recognizes double-stranded RNA as being foreign, and it processes that for degradation, and that results in any homologous genes being silenced. So any pathogen messenger RNA that has the same sequence as the double-stranded RNA that we apply, or any viral RNA, will get degraded. And this results in protection on the plant. And the beauty of this approach is that it's highly specific. So we can use bioinformatics to make sure that we're only targeting the specific pest or pathogen, and that we're not going to accidentally target a beneficial organism. There's no chemical residues that are left on the plants. And if the pathogen did develop resistance, it's very easy to deploy a new double-stranded RNA against that pest or pathogen. And this is all non-GM, so nothing is being integrated into the host plant. And if you're not aware, in 2019, Australian Parliament actually updated their GMO guidelines or regulations just to make sure that this exogenous double-stranded RNA application is exempt. So basically, as long as the RNA cannot be translated into a protein, it doesn't become integrated into the organism's genome, and it doesn't give rise to any infectious agent, then this technology is considered non-GM. There are some limitations with spraying naked double-stranded RNA onto plants, though. RNA is quite unstable in the environment, so it can degrade when it's on a plant surface. It can also wash off if it rains, and this means that there's a very short protection window. However, Nina Mitter, together with her lab and collaborators at the AIBN, has developed a product known as BioClay. And so this consists of the double-stranded RNA, which is the active ingredient targeting the respective pest or pathogen, bound to clay particles. And the clay acts as the delivery agent, and it actually protects the double-stranded RNA from degradation. And so what happens is that we spray this BioClay onto the plants, and the clay just naturally degrades over time, gradually releasing the RNA onto the plant. And so this extends the protection window because there's a very slow release of the double-stranded RNA onto the plant surface. And so these results sort of show that. So this is a plot comparing naked double-stranded RNA on leaves to the BioClay. And you can see that the RNA, when it's bound to the clay, is still intact on the leaves 30 days after application, whereas the naked RNA has started degrading at 10 days. And this result is showing a rain simulation. So when we washed with water, you can see that after washing, the naked RNA is completely removed from the leaf. but the RNA in the bioclay is still present. And the RNA present in the bioclay can protect against viruses up to 20 days after it's applied. And so, yeah, this schematic is just sort of showing the procedure. And these are some examples, just so you can get an idea. So this poor plant has been infected with tomato spotted wilt virus. It's not looking very happy. This plant was sprayed with the bioclay, which contained the RNA targeting that virus. And you can see it looks very healthy. These are some close-ups of the leaves. And this is a field example against zucchini yellow mosaic virus. And once again, the virus-infected plants don't look very healthy. When the bioclay is used, the plants are protected from the virus. And so this method has also been translated into protection against insect pests. So Ritesh just recently published this paper on using bioclay to target whiteflies. And so this result is just showing that when he actually soaked leaves in double-stranded RNA and then put some whiteflies on the leaves, the whiteflies actually took up double-stranded RNA through the leaf. And so this image is showing fluorescently labeled double-stranded RNA. It's labeled with Sci3, and he was able to see it within the insects. So we're able to do some quite cool things with this double-stranded RNA technology. And what we wanted to know is whether we can go one step further and use exogenous RNAi to control against fungal and oomycete pathogens. And so these are some of the pathogens that we're interested in and that we've been working on. So Botrytis cinerea, which causes grey mold. You might have seen it on your own strawberries. If you've left them in the fridge for too long, they get this nice furry kind of covering on them. Colototricum, which causes anthracnose, and this is showing anthracnose on an avocado. Ostropaccinia sidii, which causes myrtle rust. And so this disease actually infects a lot of our native Myrtaceae species. And then these two are actually root pathogens, Phytophthora cinnamomi, which is an oomycete, and it causes Phytophthora root rot. This is showing Phytophthora on some pineapples. And Verticillium dahliae, which causes Verticillium wilt. And this is showing cotton, I think. And so this is also one of the, well, it is the focus of the ARC Hub for Sustainable Crop Protection. So I have been working a bit with the hub on these diseases. And this is a huge consortium which consists of a lot of researchers from across Australia, also from America, and some of our RDCs. And so the idea of the hub is basically to sort of target specific economically important fungal diseases and try and commercialize a bioclay product. So going from the whole process of identifying target genes, synthesizing, doing laboratory tests, and then scaling it up into the glasshouse in the field, but also looking into registration and social licensing. So the hub is also supporting some of the work that I'm doing and sort of like providing information which is helping me in my project. So examples from the literature. So these were some of the first examples of people targeting fungal pathogens. So this example is showing control of sclerotinia on canola. And so they tested 3 different RNAs targeting sclerotinia. And as you can see, compared to the control, which was just water, the lesions have been reduced quite significantly on the leaves. And this example is showing control of Fusarium graminearum on barley leaves. So in the non-treated leaves, these lesions develop, but when they targeted a Fusarium gene, they found protection against the pathogen. And this example is from Hailing Jin's group in the US, who we've also been collaborating with. and she demonstrated protection against Botrytis cinerea on various fruits, on leaves, and also on flowers. So what we wanted to know first is whether the different fungal pathogens that we were working with were actually able to take up double-stranded RNA from the environment. And what we found is that most of them could take up double-stranded RNA, but with different efficiencies. So the rust fungi and Botrytis were really efficient at taking up double-stranded RNA. So nearly all of the germinating spores displayed this fluorescence when we applied the Cy3 labeled double-stranded RNA, so the fluorescently labeled double-stranded RNA. Whereas for pathogens like Verticillium and Colletotricum, we saw fairly limited RNA uptake, and it seemed to be very localized. So we only saw fluorescence from certain regions of the fungi. So this is just something that we have to keep in mind when we're conducting our experiments and designing our RNAi constructs. And another interesting thing we found is that the RNA uptake also differs depending on the life cycle stage. So this is showing a few different Phytophthora species. So Phytophthora is the oromycete pathogen that I'm looking at. There was quite patchy uptake into mycelia, but really nice uptake into the developing sporangia, as you can see here. And the sporangium actually is where the zoospores develop, and these are the infectious spores that then swim towards plant roots and infect the roots. So it looks like the zoospores take up the double-stranded RNA when they're in the sporangia, but I also tried adding double-stranded RNA to isolated zoospores, and they didn't seem to take up the RNA. So we need to be quite careful as to what life cycle stage we're targeting here. So we first started looking at Botrytis, seeing as this group in the US had already done some work on Botrytis. So we formed a collaboration with their lab at the University of California, Riverside. And so Jonathan was a postdoc in the lab at the time, and he set up a lot of experiments on tomato plants. They had a nice assay set up where they sprayed the plants with the double-stranded RNA or the bioclay. And then they harvested the plants, and at different time points, they inoculated them with the Botrytis fungi. And so this is just showing the results with the double-stranded RNA, but this work has now been conducted with the bioclay as well. And what we've found is that when we target specific Botrytis genes, we do see a reduction in the lesion sizes on the tomato leaves. So it's looking quite promising. And he also has conducted some post-harvest disease assays. So he's mostly been working on tomato fruit. This is from one of their earlier papers where they also conducted experiments on leaves, petals, and grapes. And once again, when the specific Botrytis double-stranded RNAs were targeted, there was a reduction in the mold development or the lesions on the fruit or the leaves. And I just want to mention as well that we've always been including non-specific RNA controls in our experiments. So in this case, it was YFP. And this shows that it is a sequence-specific effect. So we really do need the Botrytis-specific RNA to see a reduction in the lesion sizes. The non-specific RNA didn't have any effect. And so we've also been collaborating with some researchers at Griffith University to look at whether we can use RNA and bioclay to protect chickpeas from Botrytis cinerea. And so Sam from Griffith has been conducting these whole plant assays on chickpeas. And we've also seen some nice results from this. So when we treated the plants with RNA and bioclay, you can see they're looking a lot healthier. And this is up to 1 month after infection. Compared to the non-treated plants, which sort of totally wither up and they're all brown, and this is all necrosis from the fungus. So these results are looking really promising. So hopefully we'll be able to scale up soon. So as I mentioned, some of the other pathogens I've been targeting include Colletotricum on avocado. So for Colletotricum, we're looking at sort of the post-harvest disease. So I conducted some assays on avocado fruit. The in vitro results were promising. I did see some reduction in fungal growth on agar plates. However, when I did the fruit assay, I didn't really see any difference in the lesion size. So I might have to look into this further. One of the more promising results was for Phytophthora cinnamomi on pineapple. And so this work was also conducted together with researchers at DAF, and also my PhD student Lenny Palmer is helping with some of this. So in this experiment, we grew the pineapples hydroponically so that we could look at the roots and we didn't have to dig them out of the soil. So what we did is that we applied RNA into the water that the roots were growing in, and then we inoculated them with Phytophthora. And then we looked to see how the disease progressed. So with the RNA, you can see that the roots are white and they're quite healthy. Whereas when we didn't have any RNA present, the roots developed these brown lesions. And this is actually not too bad an example. They can get much worse. So this was quite promising. But now we're looking into different application methods, like whether we can apply the RNA to the leaves, because applying it to the roots in hydroponics isn't so practical for growers. And so now I wanted to spend some time on some of our results against rust fungi. And we're hoping to publish this work very soon. And so this is work led by my PhD student, Beck Degnan, together with Alistair McTarget and Louise Shuey, who's also at DAF. So Myrtle Rust is a huge problem in Australia. It arrived in 2010, and it arrived in New South Wales, and it's since spread pretty much all across Australia. And it poses a really serious threat to our native biodiversity. And this is because it can infect more than 350 of our native Myrtaceae species. So it can have a huge effect on our natural ecosystems. And it's spread in the wind, so it's windborne. So that's why it's been spreading so quickly. And it also infects new growth. So the problem is that when plants put out their new growth, this is the growth that gets infected, unfortunately. And symptoms, so this is sort of the typical symptom. You see these yellow pustules. You might have already seen this on some plants in your garden. So it can sort of range from this to whole tree malformation and mortality. So we wanted to see whether we could develop an RNA interference-based control for Myrtle Rust, but we were also looking at other rust fungi in general, because they're the largest group of plant pathogens. So the other rust fungi we've been doing some work on is Polyosporium Plumeria, which is the fungus that causes Myrtle Rust, which you've probably also seen around. So we included this in some of our experiments as well. So first of all, we did an RNA uptake study, once again using the Sci3 labeled double-strand RNA, which fluoresces. So this is a mixture of two different types of spores, uridinospores and teleospores. What we found is that the uridinospores could take up the RNA really well. So these are the infectious spores that cause those yellow pustules that I just showed you, whereas the teleospores didn't take it up. So the teleospores are more of a resting spore, and perhaps that's the reason they didn't take them up. And we also saw uptake in frangipani rust in the uridinospores. So that's quite promising that the uridinospores can take up double-stranded RNA. Interestingly, it looks like they take it up quite early on in germination. So you can see here the germ pores have just started developing, and they've already started fluorescing with the double-stranded RNA. Again, you can see here the developing germ tube. So it looks like the RNA is taken up really early, and we can kind of confirm that in these experiments. Here we actually targeted rust-specific genes, and we found that the rust-specific RNA actually inhibited germination. So this is with a GFP double-stranded RNA. This is all on an artificial leaf surface, so mimicking a leaf. And so this is the Aprosorium and infection peg, which are the infection structures that start developing once the spore germinates. And they're the structures that are used to penetrate into the leaf. So this is kind of what normal spore germination looks like. And then when we added the RNA, well, first of all, not all of the spores germinated. So there was a reduction in spore germination. But for the spores that did germinate, they actually didn't form these defined infection structures. So the closest we saw was a slight swelling at the end of these germ tubes. So it looks like when we target these rust-specific genes, we can actually inhibit spore germination. And so this top line is all Osteopuxinia acidii, and the bottom line is all the Polyosporium Plumeria. So we saw the same thing in 2 different rust fungi. So we then took this onto a leaf surface. So Beck conducted all of these detached leaf assays. We screened a whole heap of double-stranded RNAs targeting Osteopuxinia acidii. And so most of these targets worked pretty well. So on the left in each of these leaf photos is the control. So that's where we just have spores mixed with water pipetted onto the leaf. And then on the right-hand side of each image here, it's the rust-specific double-stranded RNA. So spores mixed with the double-stranded RNA. So in all of these cases, for all of these different RNA treatments, we saw a dramatic reduction in pustule formation. It's quite clear to see on these leaves. Some of the targets weren't as successful, and the GFP, so the non-specific RNA, you can see there was no difference. So the pustules developed regardless of whether there was RNA present or not. So we selected some of the most successful constructs, and we performed assays on whole plants. So these were on 1-year Syzygium jambos trees. I should say in this assay, it was also Syzygium jambos leaves we were using. This is actually not a native Myrtaceae species, but it's highly susceptible to Myrtle Rust. So it's a good model to use to test our RNA. And so once again, when we applied the GFP double-stranded RNA, pustules developed on the leaves. So these photos were taken 2 weeks after we applied the inoculum mixed with the double-stranded RNA. This is the water control. And for the 2 RNA treatments we tested, we really saw minimal symptoms. It was hard to see many pustules at all. So these results were really promising. And so then we started playing around with the timing of the RNA application. So we tested applying the RNA 6 days before we inoculated the plants. And once again, we saw protection. So this is showing without any RNA, the pustules developing. And this was the plant that had the RNA applied. And so that made us think, well, can we also apply double-stranded RNA curatively, so post-inoculation? And so Beck has now developed this whole kind of flow system where it's kind of like a continuous experiment. So she can apply the RNA at different time points, either repeated RNA sort of doses or just one dose at a certain time point. But these are kind of the time points that we're testing. So we've tested 24 hours post-infection. So that's when the spore should have penetrated into the leaf. It should have germinated. 6 days post-infection, which is when you see the first symptoms on the leaves. 8 days post-infection, which is when you see the first pustules developing. and then much later on in the infection, so more of, at the point where the infection is established, so 14 to 19 days post-infection. So we're testing all of these different time points to see whether the RNA will be able to cure the plants as well as prevent infection. And there's also different time points for the preventative treatments. So we tried 6 days. We've also tried 48 hours, and we could even try earlier. And so very excitingly, yes, it does seem that we can apply the RNA after inoculation and still protect plants from disease. So in this photo or this image, it's showing an experiment where Beck applied the double-stranded RNA 6 days after inoculation, which is when you can first see disease symptoms, which is sort of like, you see here, there's sort of like red marks on the leaves start. So basically, the disease didn't progress once she added the double-stranded RNA, which is pretty interesting. So now we're also looking at the mechanism of like what's happening in the plant. How is this actually all working? And again, this is sort of just showing all of her plants that, well, these ones are without RNA and these are with the RNA. So this is actually 6 weeks after inoculation. And these were the different time points she was testing. So she was applying the RNA curatively post-infection, and then just assaying the plant health. So overall, plant health had improved dramatically with the RNA compared to without the RNA. They were looking quite sick and unhealthy. So the success we've had with the Myrtle Rust has now made us think, well, is it something to do with rust fungi? Are they really amenable to exogenous RNAi? So now we're starting to look at some other rusts. So for example, blueberry rust and a few of the wheat rusts, so wheat leaf rust and wheat stem rust. And Alistair has actually done a lot of phylogenetics to see whether the RNAi genes are conserved across rust fungi, and indeed it does look like they are quite conserved. So we were able to identify dicer-like proteins, the RDRPs, so the RNA-dependent RNA polymerases, and the Argonauts. So all of the proteins required for the RNA interference mechanism, they do seem to be conserved across rust fungi. So that's quite promising. So hopefully we can target other rust fungi with RNAi. And so the other sort of parts of the project, and so for my Advanced Queensland project as well, we're sort of looking now at taking this out of the lab and into the field. So how can we facilitate adoption of this technology? So something that's really important is how we're going to deliver RNA to the plants. And this is obviously going to differ depending on the host plant, because different plants have different physiologies, different types of leaves. So we've been looking at the different sort of host plants and how we can deliver RNA to them. We're also looking at the longevity of disease protection. So we'll be testing bioclay to see if it can help extend the protection window. And then hopefully we'll move into the field. And so this is with the support of all of these industry partners. So this is kind of some of the ways we're looking at applying RNA to the plants. So for pineapples, we've obviously applied the RNA directly to the roots through the hydroponic solution, but now we're also looking at crown dips. This is how growers do apply fungicides in the field. So basically these crowns are dipped into fungicides before they're planted. So we're testing whether we can actually dip the whole crown into the double-stranded RNA or the bioclay, and then whether that protects against disease. We're looking at RNA uptake by pineapple leaves to see if that's a way to get the RNA into the plants. For the avocados and the myrtaceae, we're doing foliar sprays. We're also applying RNA to the petioles. So we're cutting off some of the branches and dipping the petioles in the RNA. And we're also injecting RNA into larger trees. So this has been done for avocado and Syzygium jambos. And why we're looking at all of these mechanisms is that, so Chris Brosnan from Nina's lab has done some work on Verticillium dahliae and Fusarium oxysporum. And he's shown that he can actually apply double-stranded RNA to leaves. So this example is Arabidopsis, but he's also shown it in other host plants. And that RNA can actually move down into the roots and into the infecting pathogen. So the idea is that we don't have to directly apply the RNA to the roots. We could apply it to the leaves or inject it into the trunk, and then it can move systemically throughout the plant. So that's what we're testing at the moment on these other host plants. But he actually saw a knockdown when he targeted GFP in Verticillium dahliae and Fusarium oxysporum. So it looks like RNA that can be applied on the leaves is still functional by the time it gets into the fungal pathogen. So that's pretty exciting. So for the pineapples, like I said, we're looking at applying the RNA into the crowns. The crown is actually a reservoir for water and nutrient solutions in pineapples. But nutrients are also absorbed through the roots, but also the basal white tissue, which isn't shown in this photo. The only problem with pineapples is the leaves are quite tough. They're cam plants, so they're sort of like have these tough leaves with like few stomata on the leaves to conserve water. So it's going to be quite tricky for the tomatoes. We tried an experiment with food dye where we wanted to see whether the dye would actually get into the plant when we applied it to the crown, and it didn't move at all. And the same thing when we dipped the roots in the dye, it didn't actually go further up the plant. So now we're looking at using different adjuvants to see if we can get the RNA in through the crown, or otherwise, we'll have to do some kind of soil application. But delivery is a bit easier for the myrtaceae species. So we've done some leaf applications and we've also soaked petioles in the double-strand RNA. So this is for two species, Syzygium jambos, but also for lemon myrtle. So what we found is that when we apply RNA to leaves, it just seems to stay on the sprayed leaf. It doesn't move into young leaves. But when we apply the RNA to petioles, it is detected in the new growth. So it looks like we can apply it through the petioles and it will move into the new leaves. And this was all RNA that was extracted from plants 1 week after applying the RNA. And so that's why now we're also looking at doing trunk injection experiments for these bigger trees. I'll show you this video. We've had to actually drill holes in the trunks of these trees because they're quite thick. And then once we've got the hole, we actually insert this syringe in and inject the RNA into the trunks. So really excitingly, we actually detected intact double-stranded RNA in the new leaves and the roots 6 weeks after we delivered it. So this is for a spray application, but also from a trunk injection. So these are 3 plants where we injected the RNA into them as well, and 3 other plants that we sprayed the RNA, looking at the leaves and the roots. And so we could actually detect the RNA in the leaves and the roots 6 weeks post-application. There was still quite a lot of RNA present on the sprayed leaves though. So this is quite promising. And so this is all for avocado, but I've now repeated this experiment for the metacees. So I'll see if we see a similar result. Oops, sorry. So yeah, that's the end of my talk. I'd just like to thank everyone. So everyone in Nina's lab, everyone in Bernie's lab, but specifically these people here. So Beck did a lot of, she did most of the Myrtle Rust work. And Lenny's doing now most of the Phytophthora work. My collaborators at DAF, and also our collaborators from New Zealand who actually designed the artificial leaf that we were using for all of our spore germination experiments. And also I'd like to thank Advanced Queensland for funding my research and all of these industry partners who are supporting me in my research. Thank you. And I'll answer questions.
Yeah, thanks for an interesting seminar, Anne. Now you can see the Q&A, can you? There's one in already, so maybe I'll just leave that to you. Perhaps just repeat the question so that the rest of the audience can hear.
Okay, so the question is, when you spoke about the whiteflies integrating the double-stranded RNA, it got me thinking about agricultural application for this method. Is there any worry about the double-stranded RNA affecting the human microbiome if it was on produce? So I guess that's a question about off-target effects. And so we are trying to make sure we check for off-target effects. We put all of our sequences through a software that will actually make sure that there's no sequence homology with any beneficial organisms. With the human microbiome, I'm not really sure. It shouldn't have any effect on bacteria. I'm not sure about other microbes, but we're really trying to make this very specific so it shouldn't have any effect. And it does degrade quite quickly too. So there shouldn't actually be any residues left on the produce by the time you come to eat them, I wouldn't think. There's another question. Do you want me to read that one out?
Yeah, go for it. Yep.
How long is the first bioclay RNA away from market? I think it's still quite a fair way away from being on the market. There's quite a lot that has to happen first. So more field trials, and then it has to be scaled up and commercialized. So yeah, I don't know, Don, if you have a better answer for that.
I can step in there. I think we can take out the bioclay component, but there is a Colorado potato beetle RNA product that a company in the US is looking to launch in 2023. It's gone through quite a few field trials and going through registration at the moment. So that's a anti-insect RNA molecule. Whether it gets through that hurdle and how that holds up against commercial insecticides, I guess we wait to see, but certainly pretty exciting in the RNA front anyway. So have we still got time? Yeah, we've still got a few minutes. I've got a question. One of the things that strikes me about the RNAi space is that in some cases it's going to be really, really effective, and then some organisms aren't quite as susceptible, if you like. So how are we going to, I guess, change the expectations if RNAi is supposed to be the thing that's not as effective as our existing control mechanisms like fungicides?
Yeah, I guess this is maybe not going to be used in isolation. So it'll be part of an existing integrated pest management strategy. So it might not completely replace fungicides or pesticides, but hopefully it will be able to reduce their usage. So it might be able to be applied alongside them or along with other methods. But yeah, it depends. If it's very successful, like for Myrtle Rust, maybe it will completely replace fungicides. That would be great. But for some of these other pathogens, it might sort of just be part of the strategy.
Yep. And there's a question from Tim here about evolving resistance to RNAi.
Okay. Are there any known examples of target pathogens developing resistance to RNAi vectors or hints of it? Not that I know of. I think for resistance to develop, they'd have to actually lose part of their RNAi mechanism, like the core machinery would have to somehow mutate, and that probably wouldn't happen seeing as it is so essential. It's not just, it's sort of a very essential mechanism. So I haven't heard of anything.
There is in the insect space.
In the insect.
What we don't know there is what effect that has on the survivability of the insect. So you can induce it in the lab, but how that plays out in the ecology of that organism, we don't know.
Okay. Yeah. So we're not really sure. No, that's the answer, I guess. Thanks. Sorry, he's just clarified target-based. So based on like the essential, well, we are trying to target essential genes. We're not really targeting effectors. So they're sort of different targets to what a normal fungicide or a pesticide would be targeting. So we're sort of assuming resistance won't develop because they're sort of essential genes that are required for growth and development, not pathogenicity. So will this work against multiple pests/insects? Would this include multiple RNAi, and would this affect the efficiency? So for insects, there's differences in efficiency depending on the type of insect. So some work better than others. Multiple RNAi. I guess like we're trying to target viruses and their insect vectors in some cases at the same time. So stacking different RNAs, so targeting the virus, but also the insect that transmit the virus. So there are lots of different options, I guess, for insect pest control. I'm just reading the second question. Have we faced any backlash or hesitancy from people for using GM in food products? Well, our method is not GM, so we're trying to avoid that. People are worried about GM, I guess, just because they don't really understand it. So we're sort of trying to avoid that whole sort of question or debate, but we are still worried that people will be cautious about RNA because that's also something a bit scary. Although I think now with all these, COVID vaccines that are based on RNA, hopefully people are a bit more open to us using RNA.
I'm going to follow up there, Anne, with one more question around RNAi and medical use. I mean, you mentioned that some of the oomycetes might be able to be targeted. Would RNAi be useful in any sort of control of human pathogens, let's say, or I'm thinking malaria or something like that?
Yeah, I guess so. Maybe if it's used against mosquitoes that transmit the malaria. And I mean, people in our lab are extending the work into animals. So Karishma is working on fire ants and sheep blowflies. So it's not just limited to plant pests. It can also be animal pests and pathogens, and possibly human ones, I guess, if it's vectored by an insect. But yeah, there are sort of like numerous possibilities, I think, with this technology. It doesn't have to just be plants, but obviously we're interested in plants.
Okay, I think perhaps we'll leave it there. There's no more questions on the Q&A. So thanks very much for attending. Just in closing, I'd like to just make you aware of the next talk, which we have coming up next week. I think that date is. So we'll be hearing from Andrew Borrell, and you can see the information on the slide there. So thanks everyone, and thanks very much to Anne for a great seminar.